Okay, bad jokes aside, today we'll be looking at Pasteurella multocida in cattle. We'll be looking into its classification, its characteristic features, how the organism is contracted and spread, whether the organism can survive outside and/or inside the host, the common predisposing factors for infection, as well as which types of samples you will take to send to a laboratory and how these samples will be preserved and transported. Lastly, we will compare this organism with other bacteria, viruses, and fungi. We are Group 23, and now let's meet the members. Tanushka Gounden, Jenna Lewis, Nicholas Lonsdale, Kira Lee Manny, Claire van Niekerk, and myself, Tanya Smith. Now, unlike this group, our group demonstrated good communication. We divided the work equally amongst ourselves and we helped each other when needed. Now, let's get back to P. multocida. Pasteurella multocida is classified as a pathogenic bacterium. Here we can see a list of its characteristic features, namely that it is gram-negative, non-motile, non-spore-forming, a cocobacillus, it has bipolar staining features, is penicillin-sensitive, belongs to the Pasteurelliaceae family. It is a facultative anaerobe, is oxidase and catalase positive, and can ferment carbohydrates in anaerobic conditions. Different strains of this species are classified into 5 serogroups, A to F. P. multocida exists as a prevalent commensal in the upper respiratory tract of many animals. Do animals contract P. multocida? Yes, P. multocida is the cause of disease in many animals, for example livestock, where we will be focusing on cattle. P. multocida can cause pneumonic pasteurellosis in cattle, which is an acute pneumonia which forms part of the cattle respiratory disease complex. This organism is part of the normal upper respiratory microflora of cattle, and it mostly only causes disease when an animal is already compromised. It greatly affects cattle because they have a small internal lung surface area per body weight. Thus, they are unable to compensate if a part of their lung is compromised due to infection. How does this pathogen spread? It is an acute airborne disease that spreads when infected animals cough or sneeze, transferring infected droplets to other animals within the vicinity. Again, the spread can be enhanced by certain predisposing factors like stresses, dust, and vaccinations. This diagram just illustrates the infection cycle as discussed in the previous slide. Where can P. multocida survive? It is able to survive inside the host as well as in the environment outside of the host. Virulence mechanisms such as capsules assist this pathogen to survive the internal environment of the bovine host. For the outside environment, the lifespan of P. multocida varies according to the different surfaces and mediums within the environment. It can survive for approximately 14 days in low temperature water, contrasting its survival rate of only approximately 45 minutes in the air. We already named a few of the common predisposing factors for infection, but here is a recap. The factors include stressors, high daily ambient temperature fluctuation, noxious gases, dust, vaccinations, infectious agents, and lastly, poor nutrition. Samples for Pasteurella multocida will be taken from the respiratory tract for bacterial culture and serotyping of isolates, such as samples of affected lung tissue, tracheal swabs and tracheal wash, transtracheal aspirates, nasal swabs for culture of infectious agents, and blood samples for serum harvest. It is recommended to take a wide variety of samples at multiple times in the year. After collecting these samples, the transport and method of preservation is important. Some tissue samples need to be put into formalin or other liquids, while other samples need to be stored at certain temperatures. For example, lung tissue samples should be fixed in 10% neutral formalin and embedded in paraffin by routine methods before transport. Nasal swab samples should be placed on dry ice and subsequently be transported in -70°C freezers. These samples are frozen for isolation, identification, and quantification of viruses and bacteria. All samples should be concealed correctly, labeled clearly and with the correct information, and then transported with great caution. If you are unsure of preservation or transport protocols required for a specific sample, it is best to call the laboratory and find this information out beforehand. How does Pasteurella multocida compare to other bacteria, viruses, and fungi? P. multocida is a pathogenic bacterium from the class Alpha Proteobacteria. In the following 3 slides, a comparison table has been made to compare these different organisms. Looking at morphological characteristics, we can see that P. multocida is an average size of 1 to 2 micrometers compared to other bacteria. It is Coccobacillus in shape with no flagella. Viruses are a lot smaller than our bacteria, ranging from 20 to 400 nanometers. And fungi are usually bigger, ranging from 2 to 10 micrometers. For reproduction, P. multocida reproduces asexually through binary fusion, but other bacteria can use other methods like budding as well as sexual reproduction methods. Viruses use lytic cycles and/or lysogenic cycles, while fungi reproduce asexually by budding, fragmentation, and spore formation, as well as a few sexual methods. Looking at genetic exchange, in P. multocida, bacteriophage F108 performs transduction, but conjugation and transformation can also take place. Other bacteria also use conjugation and/or transformation for genetic exchange. Viruses take a piece of DNA from the host cell and incorporate it into its own genome. Fungi use horizontal gene transfer. For laboratory identification techniques, PCR, microscopic cultures, and specific stains are used for P. multocida. For other bacteria, you can use staining, cultural characteristics, biochemical techniques, immunological techniques, phage typing, and/or molecular techniques. For viruses, one can also use molecular techniques, often followed by secondary nucleic acid tests to confirm positive serological results. For fungi, serological tests like ELISA is used. Sometimes molecular techniques will be used. Here is a list of the references that were used. Oh, sorry. Last one, I promised. Thank you for your time.